Sorry, I was just telling that normally we wait 5 minutes for everybody to come, but in the meanwhile I can just scientist and professor in the Department of Biomedical Sciences at Colorado State. She in neuroscience at Washington University School of Medicine in St. Louis, Missouri. After this, she went to postdoctoral work at UCSD and had her first faculty position in the Department of Biology at Boston University. Dr. Tsunoda has been a principal investigator and faculty member in CSU since 2009. And her work has been dedicated to the study of ion channels and receptors, their regulation, organization, trafficking, and function. All of her lab's studies use Drosophila melanogaster as a model, combining genetics, cell biology, biochemistry, and electrophysiological techniques to study the function and plasticity of neuronal signaling. She has trained 11 PhD students, 5 postdoctoral fellows, and more than 25 undergraduate students. From Dr. Tsunoda's lab have been published in well-respected journals, including one of the last she has published in PLOS Genetics, Nature Neuroscience, Nature Science, PNA Procedures of National Academy of Sciences, and the Journal of Neuroscience. Dr. Tsunoda has always been passionate about supporting women, minority, international, and underrepresented students in science, and started her independent career as a Clark Booty Luke Assistant Professor for Women in Science and Engineer at Boston University. And she has served on the College Founders Committee for Diversity and Inclusion at CSU. Her lab has been filled with trainees from around the world: Ethiopia, Libya, Saudi Arabia, Korea, India, China. Dr. Tsunoda has taught a variety of cell and molecular neuroscience courses over the years, and currently teaches a course she created that is open to all undergraduate students to try out research for a semester. Dr. Tsunoda is also a new director of the Molecular, Cellular, and Integrative Neuroscience program at CSU for both undergraduate and PhD students. And we welcome her. We're very happy to hear her thoughts. I want you just to remind every student that there will be this opportunity to meet with her at lunchtime to have a closer conversation to her about her career and science, to questions, everything. So after the presentation, we wait for you in the first floor meeting room. Thank you.
Okay. Good afternoon, everyone. I'd like to thank Dr. Moreno for the opportunity to share our work with you. And I hope to hear about what all of you are doing, maybe over lunch. So I'm going to talk about today is homeostatic plasticity in the brain. So homeostasis is a self-regulating system or process that maintains stability and adjusts to changing conditions. And of course, plasticity is the ability to exert those changes. So you can think of homeostasis sort of like a thermostat in your house or a building that maintains a particular temperature. And when the temperature changes and becomes hotter or colder, the thermostat senses those changes and regulates heating and cooling systems to maintain and counteract those changes and maintain a particular set temperature. So similarly, in our bodies, a lot of essential physiological systems are also under homeostatic control, including core body temperature, blood pressure, blood glucose levels, blood oxygen and CO2 levels, the volume of body water, extracellular sodium and potassium concentrations, as well as neuronal activity. And that's what I'll be talking about today. So the brain and neuronal activity are also under homeostatic control. In the human brain, there's about 86 billion neurons, and those neurons communicate with one another, forming circuits in the brain and also communicating with the periphery And they communicate with one another through these structures called synapses, which I probably don't have to describe in this audience. But these synapses is communication where a presynaptic cell communicates with a postsynaptic cell. In the presynaptic cell, there's an electrical signal that's made up of, that consists of a fast transient increase in membrane potential that's called the action potential. And these action potentials travel down the axon of the presynaptic cell to the nerve terminal of the presynaptic cell, where they signal the release of vesicles containing neurotransmitter. Neurotransmitter is released into the synaptic cleft and received by receptors on the postsynaptic membrane. So this is how, and these, the activation of these postsynaptic receptors then signal membrane depolarizations in the postsynaptic cell. So this is how the presynaptic cell communicates with the downstream postsynaptic cell at these structures called the synapse. And the activity at these synapses is what is under homeostatic control that I'll talk about today. So neurons in the brain encounter changes in activity both during physiological as well as pathological conditions. So during development, there are well-documented instances when there's a change in activity that is necessary for development. During learning and memory processes, of course, there are Hebbian plasticity processes that occur. And also during different pathological conditions, in these diseases and disorders, including epilepsy, Alzheimer's disease, Parkinson's, traumatic brain injury, stroke, and ALS, it's been increasingly found that there are large changes in neuronal activity. So when neuronal activity is changed, neurons respond by increasing or decreasing their synaptic strength to counteract those changes. And this is what we call synaptic homeostasis. So synaptic homeostasis is sort of like a thermostat for neuronal activity. This is a diagram of a synapse. This is the presynaptic cell on the top and the postsynaptic cell down below, and it's releasing these vesicles of neurotransmitter to the postsynaptic side. When the brain encounters overall reduced network activity or decrease in global activity, we see that synapses will do things to increase or boost activity. And shown here is, for example, an increase in neurotransmitter release or an increase in receptors on the postsynaptic membrane. Conversely, if the brain encounters increased network activity, for example, during seizure, neurons and synapses will do things to downregulate or decrease synaptic strength. And here shown is a decrease in receptors on the postsynaptic membrane. So synaptic homeostasis is classically studied by imposing a decrease in neuronal activity and then studying how those synapses increase or boost synaptic strength, and that's called synaptic homeostasis. I'll also show you that, that there are also regulatory mechanisms that are being discovered that help to prevent overcompensation by these homeostatic mechanisms and help to then stabilize neuronal activity in an optimal range. So how do we measure synaptic activity and know when it's increased or decreased? And we do that by recording miniature excitatory postsynaptic currents, or miniature EPSCs, as an assay for synaptic activity. So a miniature EPSC is a response on the postsynaptic cell to the release of a single vesicle of neurotransmitter. So when a single vesicle of neurotransmitter is released, it activates a small number of receptors on the postsynaptic membrane, and these receptors are then opened and they conduct a sodium and calcium current into the cell. And these currents can be recorded by an electrode in the postsynaptic cell. Those signals are amplified, and we record them as miniature EPSCs. So they look something like this. Here's the baseline, and each of these downward deflections is an individual miniature EPSC. When one of these is expanded, you can see it looks something like this. And because it's the response to a single vesicle of neurotransmitter being released, if we see a change in the frequency of those miniature EPSCs, it's thought to support the idea that there's a change on the presynaptic side, for example, an increase or a decrease in neurotransmitter release. If we see a change in the amplitude of these miniature EPSCs or the shape of these miniature EPSCs, it's consistent with the idea that there might be a change on the postsynaptic side, for example, an increase or decrease in the number of receptors. Okay, so this is what synaptic homeostasis looks like at glutamatergic synapses. So this is a control trace, And these small downward deflections are miniature EPSCs. And classically, we study synaptic homeostasis by blocking activity. And so in this case, researchers used an antagonist against all glutamate receptors called NBQX. They block receptor synaptic activity, and then after a prolonged period of time, they wash out that antagonist, and then look at what the miniature EPSCs look like after that. And you can see here that now the miniature EPSCs are both more frequent and larger in size. And so that's the homeostatic response, and it indicates that there's both changes on the presynaptic side because there's more frequent miniature EPSCs, as well as changes on the postsynaptic side because these responses are larger. And over the decades, the last 2 decades, there's been a lot of different pathways that have been identified, both on the presynaptic side and the postsynaptic side, that are responsible for these changes in miniature EPSCs. And most of the studies on synaptic homeostasis have been at glutamatergic synapses, since glutamate is the primary excitatory neurotransmitter in the mammalian brain. But there are also cholinergic regions of the brain, as you know, that are important, and many of which are associated with disease. So well known, of course, is the loss of cholinergic neurons that happens in the basal forebrain in Alzheimer's disease. There's also changes in cholinergic markers and changes in acetylcholine transmission that occur and associated with many other different diseases. But not much less is known about how the receptors at these synapses, called nicotinic acetylcholine receptors, how they are affected by activity and whether they can mediate synaptic homeostasis. So nicotinic acetylcholine receptors play important physiological and pathological roles. They are ligand-gated pentamers that are made up of a combination of alpha and beta subunits. The homomeric alpha receptors and heteromeric alpha 4 beta 2 receptors are among the most abundant in the human brain. And I mention them because alpha 7 receptors are the ones that I'll talk about in our work. And they've been shown to play roles in a wide range of physiological processes, including motor activity, learning and memory, and attention. They're also associated with multiple pathologies, including nicotine addiction, familial epilepsy, depression, schizophrenia, and Alzheimer's disease. So we've been using Drosophila melanogaster, which is the fruit fly, as our model system to study synaptic homeostasis in cholinergic neurons. So we've used Drosophila as our model because it's been a genetic model system that's been studied for 100 years, and it has a wealth of genetic tools. So for example, we can use genetic tools to non-invasively manipulate activity in the brain. It's also accessible to study by multiple approaches, including biochemical, cell biological, molecular, and behavioral work. We also do a lot of electrophysiology, and so we can go all the way from the DNA, from the gene, to the whole animal behavior. And most importantly, for these studies, the central nervous system of Drosophila uses acetylcholine and nicotinic acetylcholine receptors as its primary excitatory neurotransmitter system, enabling us to use this system as a way to look at synaptic homeostasis in central cholinergic system. And the Drosophila brain has also been very well mapped and characterized. So I just take the opportunity to tell you that last month, in a series of papers in Nature, the entire fly brain connectome, every neuron in the fly brain, has been mapped. It contains 140,000 neurons. They make about 50 million synapses with one another. And many of the regions of the brain, as well as particular circuits, have been well defined and studied in areas such as vision, olfaction, attention, learning and memory, circadian rhythms, sleep. And so now we're going to use it for cholinergic synaptic homeostasis. So I'm going to tell you about our work in cholinergic synaptic homeostasis. And in the first part of the talk, I'll tell you about how alpha 7 nicotinic acetylcholine receptors can mediate synaptic homeostasis. So we began by looking at whether these receptors could mediate homeostasis at all, and we performed experiments just like with the glutamate receptor synapses. So we blocked activity in primary cultures using an agent called curare. Curare blocks all nicotinic acetylcholine receptor activity, And so here you can see that these are miniature EPSCs in the control trace. When you add curare, they're completely absent. So we block activity for a prolonged period of time. And then we wash out that curare and look at how synaptic activity has changed. And you can see here, after curare treatment, those miniature EPSCs are much more active, there's much more frequent and larger in size. And so synaptic homeostasis does seem to be able to be mediated by these nicotinic acetylcholine receptors. And that's quantitated here. So same thing, this is experiment after curare treatment, and we looked in different sets of excitatory neurons, and you can see a clear increase in miniature EPSC amplitude as well as frequency, although here I'm just showing amplitude. And that response is persistent for hours, so up to 5 hours. And this is not true in inhibitory neurons. When we look at inhibitory neurons, there's no upregulation of those miniature EPSCs. And this makes sense, right? So if the neurons encounter a large prolonged decrease in activity, the neurons in synaptic activity are doing things to boost or increase activity. So it makes sense that they would want to increase activity in excitatory neurons, but not in inhibitory neurons. Okay, when we looked at those miniature EPSCs in more detail, we found that they were not just increased in size, but they also had very different kinetics from the control miniature EPSCs. So it suggested to us that there might be not just an increase in nicotinic acetylcholine receptors, but maybe just particular nicotinic acetylcholine receptors were affected. And so we got our hands on antibodies for many of the different acetylcholine receptor subunits. And here you can see that we found that the alpha-7 subunit of nicotinic acetylcholine receptors was particularly affected. And we see a dramatic increase in that subunit, whereas we didn't see any increase in the other subunits that we looked at. And this subunit is called the alpha-7 nicotinic acetylcholine receptor. We refer to it as D-alpha-7 just because it's the Drosophila form. So we see a decrease in when we impose a decrease in neuronal activity, we see an increase in D-alpha-7 receptors that we think underlies this increase in miniature EPSCs and synaptic strength. And this is the synaptic homeostasis pathway that we've been studying. So those experiments were all done in cultured primary neurons, but our goal was to move more and more in vivo. So we wanted to use genetic mutants and see if we could find a way to non-invasively inhibit activity and see if the same phenomena occurred in the Drosophila brain. So we used these mutants called CHAT-TS2 mutants. These are mutants in a gene called choline acetyltransferase, which is the rate-limiting enzyme in the production of acetylcholine. So in these mutants, the mutation in this enzyme causes them to become non-functional when you raise the temperature. So when you increase the temperature to 30 degrees, you greatly decrease the amount of acetylcholine that's made. So we looked at the miniature EPSCs, we took these mutants and heat treated them, and you can see with increasing heat treatment, we get less and less of the miniature EPSCs. And that's quantitated here. So with increasing amounts of heat treatment, you get an increased inter-event time, meaning that there's less and less frequency of miniature EPSCs. So this validated that we could use these mutants as a way to inhibit activity just by heating those flies up. Okay, so we wanted to block activity like this, non-invasively, and then record from neurons in the intact brain. So we developed this preparation where we can take the heads of the flies, dissect out the brains, and then put them in a dish and record from GFP-labeled Kenyan cells. And the Kenyan cells that we record from are shown here. So this is the Drosophila brain, and the yellow shows the Kenyan cells. And these round things here, this is the cell bodies of the Kenyan cells, and then they project their dendrites into a structure called the calyx, and their axons into a bifurcated axonal pathways called the mushroom bodies. And it's been shown that when Kenyan cells are ablated, you completely eliminate learning and memory in the flies. So this site is very highly studied in Drosophila for learning and memory. It's also a site that's likely to show plasticity of activity changes. Okay, so this is the experiment we did. You take wild type or CHAT-TS2 mutants, we heat treat them at 30 degrees, which is warm for a fly, for 3 hours, and then we give them a recovery period at 18 degrees to let them recover from the block in activity. And then after that time, we remove the heads, dissect out the brains, and record miniature EPSCs in the Kenyan cells from the intact brain. Okay, and when we did this, we found that in wild type, these are the miniature EPSCs, where there's no activity blocked with heat treatment. And in the CHAT-TS2 mutants, we saw a lot more activity, and that those miniature EPSCs were both more frequent and larger in size. And you can see here, so by the decrease in inter-event time, indicating a greater frequency of miniature EPSCs, and a significant increase in the amplitude or the size of those miniature EPSCs. So again, we're blocking activity, this time non-invasively in vivo, and then we're recording from the intact brain, and we see an increase in miniature EPSC frequency and amplitude, indicating that we do see synaptic homeostasis in the intact brain. So we also wanted to see if it was the alpha-7 receptor in particular that was being upregulated in vivo as well. So here's we took flies that are expressing D-alpha-7 GFP, either in the presence or absence of the CHAT-TS2 mutant allele. We heat treat them at 30 degrees for 3 hours, give them a recovery period, and then we looked at the proteins in those heads. And you can see here that in the D-alpha-7 expressing flies, that heat treatment does nothing to the expression of D-alpha-7 GFP, but in the presence of the CHAT-TS2 allele, where we have now a block in activity, we do see a significant increase in the D-alpha-7 protein. So in vivo, we see synaptic homeostasis, blocking neuronal activity, leads to an increase in alpha-7 receptors and an increase in synaptic strength. Okay, so how are these D-alpha-7 receptors upregulated to drive synaptic homeostasis? So I'm just going to show you a little bit of data. This is closer to our current work, but we're interested in this protein in particular called NACHO. NACHO is a relatively new protein that's been identified. It's a transmembrane protein found in the endoplasmic reticulum. It's been shown to be associated with glycosylation and trafficking machinery in the ER. And it's been shown to be involved in the early assembly of nicotinic acetylcholine receptor subunits. So since it's been just recently identified and hadn't been looked at in Drosophila at all, we looked at NACHO in the Drosophila brain. This is an endogenously GFP-tagged NACHO protein in a confocal section through the posterior section of the brain. And we see that NACHO expression is pretty widespread. And it overlaps definitely with D-alpha-7 positive cells, which are shown here and marked with RFP. We also looked to see if it was likely to be found also in the endoplasmic reticulum, as reported in mammalian cells, and we see that as well. So NACHO GFP, this is probably the best one to look at. NACHO GFP is found outside of the nucleus in the cytoplasm in sort of this network-like pattern that overlaps nicely with endoplasmic reticulum markers. So we think Drosophila NACHO is also an ER protein that is found definitely in alpha-7 positive neurons. So the first thing we looked at is whether activity inhibition affects NACHO expression. So first we took our CHAT-TS2 mutants, we heat treated them, and looked at D-alpha-7 GFP first, which I showed you earlier, but this is just on a finer time scale. And this is untreated, and this is 1 hour of heat treatment, 2 hours of heat treatment, and we don't see any change in D-alpha-7 GFP levels until they're heat treated for 3 hours, and then we see a persistent increase in the D-alpha-7 GFP that lasts through these hours of recovery. So we did the same type of experiment and looked at NACHO HA, and we found that NACHO expression also seems to be activity dependent. So when we block activity, so heat treating them for an hour, we see an increase in NACHO expression, and that expression is persistent for, after 3 hours of heat treatment and different times after heat treatment. So we find that activity inhibition is inducing NACHO expression, and it seems to occur prior to D-alpha-7, the rise in D-alpha-7 expression. So 1 hour of heat treatment, you get an increase in nacho, followed by an increase in D-alpha-7. Okay, so next we wanted to see maybe, does an increase in nacho, is that sufficient to support an increase in D-alpha-7 receptors? So we used sort of this genetic trick here that I won't go into detail, but basically we can conditionally express nacho HA. And so we did that in a D-alpha-7 GFP expressing flies and compared them to D-alpha-7 expressing flies in which nacho was overexpressed. And you can see that it does lead to an increase in D-alpha-7 expression, suggesting that increasing nacho is sufficient to induce an upregulation of the D-alpha-7 receptors. If we look in the opposite direction and look at nacho expression and then look at what happens when we overexpress D-alpha-7, we see no increase in nacho protein. So it doesn't seem as though there's an increase in alpha-7 receptors that recruits nacho, but really that activity inhibition, here it is, activity inhibition is leading to an increased expression of nacho, and this underlies the increase in D-alpha-7 receptors, and that's what's supporting the increase in synaptic strength. Okay, so the question that we're working on right now is whether nacho is required for synaptic homeostasis. So if we get rid of nacho, do we still get an increase in synaptic strength that occurs in response to a decrease in neuronal activity? So we performed this experiment by creating a nacho mutant. We used CRISPR-Cas9 to knock out nacho and looked at, in this background, with and without the CHAT-TS2 mutant. We heat treat the flies for 3 hours to block activity, and then we let them recover. And then we cut their heads off, dissect the brains, and record from miniature EPSCs in the Kenyan cells in the intact brain. And this is what I showed you previously in wild type and CHAT-TS2 mutants, that we see an increase in miniature EPSC frequency and amplitude, so indicated by a decrease in inter-event time and an increase in amplitude shown here. And we did the same thing in the nacho mutant background. And you can see that with blocking activity, there's much less change in the miniature EPSCs. And that could be shown here. So the inter-event times are still a little decreased, but to a lesser extent. And the increase in amplitude is completely abolished. In fact, there's a small decrease that we're not sure why. So it seems that nacho is essential for synaptic homeostasis. It's very important, especially on the postsynaptic side, where it eliminates that synaptic homeostasis. The role of nacho, however, is complicated because nacho does play a role in the basal trafficking of nicotinic acetylcholine receptors. So currently we're doing experiments to try to interfere with the upregulation of nacho and or also affect D-alpha-7 receptors just in the residues that interact with nacho in the endoplasmic reticulum. Okay, so the summary for this part of my talk is that if we block activity, global activity, we see an increase in nacho that leads to an, increase in D-alpha-7 receptors that leads to an increase in synaptic strength. So in the second part, I'm going to tell you about how this synaptic homeostasis can also be regulated by a subsequent increase in Kv4, which is a particular potassium channel. So when we use Curare treatment, as I showed you in the beginning, to block all activity, we were also interested in, if we blocked activity, if any of the ionic currents changed. And we looked at sodium currents, calcium currents, and potassium currents. And we were particularly interested in potassium currents because my lab has for a long time studied potassium channels. These are voltage-gated potassium channels that are closed at resting membrane potential, and then with membrane depolarization, they open and extrude potassium. And so we've, in the past, been able to look in these neurons and dissect or divide the currents into two major types. There's this transient A-type current, so the current goes up, indicating an outward current in response to a voltage step. and they're rapidly activating and then rapidly inactivating. The other type of current that's present is rapidly activating and then makes the sustained outward current. And previously, we have genetically identified these currents, and it's the KV4 gene, or in Drosophila, we call it also the SHAL gene, and these underlie this channel that encodes this transient A-type current. And this delayed rectifier type current is encoded by KV2 and KV3 channels. So when we applied curare treatment to block all activity, we saw that there was no change after that curare treatment in any of these voltage-gated potassium currents. But if we wash the curare out for longer, for about an hour, we saw a selective increase, a dramatic increase in the transient A-type current that's encoded by KV4. And you can see that here too. We only saw it in excitatory neurons and not in inhibitory neurons. So this is the increase in the SHAL or KV4 amplitude that occurs after curare treatment, but only after a recovery period. So activity inhibition followed by a recovery period induces an increase in KV4 current. So just to give you a little background on KV4, KV4 is the most highly conserved of the voltage-gated potassium channels. There's a single gene in Drosophila that represents a multi-gene family in mammals. It's highly conserved in sequence, function, and localization. So at the sequence level, they're 82% identical to the vertebrate genes. All of the KV4 SHAL genes encode this type of transient A-type potassium current, and they're all localized to somatodendritic sites in neurons. And they function at subthreshold potentials. So that means they can respond to very small changes in membrane potential and are likely to play roles in modulating the postsynaptic potentials. Okay, so I've showed you that activity inhibition leads to an increase in D-alpha-7 receptors, which we think underlies the increase in miniature EPSCs, and that with some recovery after activity inhibition, we see this increase in the KV4 current. So we were wondering how they were related. And since this one occurs after this, we hypothesized that perhaps it's the increase in D-alpha-7 receptors that then leads to the increase in KV4 current. So to test this, we used a mutant of D-alpha-7. So here's the wild type SHAL or KV4 current. This is after curare treatment and recovery. It's enhanced. And if we do the same experiment in a D-alpha-7 null mutant, you can see that there's no enhancement in the KV4 current. Okay, so our working model is that blocking activity leads to an increase in NACHO, leads to an increase in D-alpha-7 receptors, and that these receptors then are triggering a secondary response, which is the increase in KV4 currents. So this, of course, is the synaptic homeostasis pathway that I've been talking about. So what's the function of this upregulation of KV4 currents that happens subsequent to synaptic homeostasis? So the increase in D-alpha-7 receptors is leading to enhanced inward current, right, to increase or boost activity in response to the block in activity. And the increase in KV4 currents is going to be an outward current that counteracts this inward current. So we think that perhaps this subsequent increase in KV4 currents its function is to modulate the inward current here and perhaps prevent overcompensation that's occurring with the D-alpha-7 receptors. And we have some data supporting this model. So we looked at miniature excitatory postsynaptic potentials. I've been showing you miniature EPSCs or currents where we clamp the membrane voltage and look at currents going across the membrane. But here we don't clamp the membrane and we look at the postsynaptic currents. And when we do that, you can see here, this is a control trace, and these are miniature EPSCs, these small upward deflections, and that whether it's curare treated and after curare treatment, those miniature EPSPs are no different from each other. So they're kept very stable. So through the mechanisms of homeostasis that I've been discussing, it's leading to stabilization of the miniature EPSPs. We wanted to see what the role of the KV4 currents was, so we used a dominant negative line that knocks out the KV4 channels. And so here in the dominant negative, you can see that those miniature EPSPs are enhanced. So in the control trace, they're also enhanced. And when you curare treat them, you see a large increase in miniature EPSP amplitude, indicating that they're not stabilized. So we think that KV4 channels are being overexpressed after synaptic homeostasis to prevent overcompensation and stabilize synaptic potentials. Okay, so what's the mechanism by which KV4 channels are upregulated? So one of the clues we had is that we know it's dependent on alpha-7 receptors, and alpha-7 receptors are known to be especially calcium permeable. So calcium comes in through the receptors and can act as a second messenger, and we thought it might play a role in the upregulation of the KV4 currents. So we performed this experiment where we mock or curare treated neurons, and this is in culture, and then recorded from the neurons during this 30-minute recovery period. And we recorded first at the beginning of that 30-minute recovery period, and then again at the end of that 30-minute recovery period. And you can see here when the cells are mock treated, there's no difference in those currents at either of these recording times, just as you would expect. But if you curare treat the neurons, you see the first recording is the black trace here, and then the second recording after 30 minutes, the trace is enhanced, so consistent still with the idea that KV4 currents are increased, but they require this recovery period. And now we are recording it in single neurons, which also enabled us to put calcium chelators in our pipette and let those flood into the cell. So if we did that experiment and we included BAPTA, which is a calcium chelator, in our pipette, we found that there was, with curare treatment, there was no enhancement of the KV4 current, suggesting that the upregulation of KV4 channels or currents is calcium dependent. Another clue that we had is that the increase in KV4 currents was also seemed to be blocked by transcriptional inhibitors. So this is, again, the controlled KV4 current. This is after curare treatment and recovery, that it's enhanced. If we use transcriptional inhibitors in the bath, we found that we saw no enhancement in the KV4 currents, suggesting that it's transcriptionally regulated. So now we wanted to use our CHAT-TS2 mutants to inhibit activity, to look at the increase in KV4, to make sure that it's happening in vivo, and also then we could get more material and look at protein levels and messenger RNA levels. So this is the CHAT-TS2 mutant with no heat treatment and heat treatment, and you can see there's a significant increase in the shell current, but not in the delayed rectifier current, so just like we saw in vitro. So CHAT-TS2 is able to mediate activity inhibition that also induces an increase in KV4 current. So now we wanted to look at protein level and messenger RNA. So we took wild type and CHAT-TS2 mutants. We heat treated them for 3 or 6 hours, allowed them to recover. And this is in wild type. You can see that with no heat treatment compared to heat treatment and recovery, there's no change in KV4 protein levels. But if we looked in the CHAT-TS2 mutants, you can see that with heat treatment and a recovery period, that there's a significant increase in the KV4 protein. So what about the RNA? Oh, sorry, this is also the protein. Sorry, this is all in the protein. So if we heat treat the flies, and here we're looking at a Finger time course, you can see also there's no significant increase in KV4 protein until after a 3-hour recovery period. So also similar to what we saw in vitro. And interestingly, that if we did the same experiment in a D-alpha-7 null background, we saw no enhancement in KV4 protein levels, also similar to the in vitro pathway. Okay, so what about the messenger RNA? So this is in wild type, and here's with heat treatment, you can see there's no change in messenger RNA of KV4. If we look in the CHAT-TS2 mutant, heat treatment induces an increase in messenger RNA of KV4, and transiently it goes up here right after heat treatment and then goes back down. If we look in the D-alpha-7 mutant, we see that we don't see this increase in KV4 messenger RNA. So activity inhibition is inducing an increase in KV4 shell, both protein and messenger RNA, and that it's dependent on d-alpha-7. So here's the pathway that we're looking at. Here's the synaptic homeostasis pathway that I've described to you, and now we see this increase in KV4 channels. And so how is this channel expression upregulated? And so far I've shown you that it's d-alpha-7 dependent, it's calcium dependent, probably calcium that's coming in through those receptors, and it seems to be transcriptionally regulated with increased levels of messenger RNA. So to look at what might be mediating that upregulation, we looked for calcium-activated transcriptional activators, and this is the candidate that we've tested. It's called NFAT. It stands for nuclear factor of activated T cells. It's been highly studied in the immune system, and more recently has been looked at in the nervous system. So NFAT is known to be calcium activated, so calcium that comes into the cell, perhaps through d-alpha-7 receptors, would bind to calmodulin, activating calcineurin, which is a phosphatase known to dephosphorylate NFAT. And when NFAT is dephosphorylated, it translocates into the nucleus and promotes transcription, and perhaps transcription of KV4. So we first had an indication that NFAT might be the transcriptional activator involved when we used this calcium reporter called Calexa, which is an NFAT-based calcium reporter. It's a mammalian NFAT that's been modified and fused to these other transcriptional factors. When it's activated, it translocates into the nucleus and can activate a reporter gene, which in this case is GFP. So we saw that in flies that expressing just Calexa, heat treatment did nothing to the expression of GFP, but if CHAT-TS2 was present, where we're blocking activity with heat treatment, we see an increase in GFP expression, indicating that this NFAT-based reporter is being activated. So to really see if NFAT is involved, we used knockout mutants for NFAT, and we looked at the CHAT-TS2 blocked activity with and without NFAT present. Heat treated the flies, let them recover, and we looked at the messenger RNA and protein. So here's just the CHAT-TS2 that I showed you earlier. The messenger RNA for KV4 goes up, is transiently increased with heat treatment, and the protein levels are also increased following the heat treatment and recovery time. When we look at the NFAT null mutants, you can see that the levels of messenger RNA increase are completely eliminated, as well as protein levels, suggesting that NFAT mutants block the inactivity-induced increase in KV4 protein and messenger RNA. Okay, so the summary of this part of the talk, I'll put it all together here, is when we block activity, we see an inactivity-induced increase in nacho protein that underlies the increase in alpha-7 receptors and an increase in synaptic strength. With the increased number of alpha-7 receptors, we think that there's increased calcium coming into the cell, and calcium activates NFAT, which translocates into the nucleus, and increased levels of KV4 expression. These KV4 channels serve to then normalize the postsynaptic potentials. Okay, in this, so that's the pathway we've been looking at. In this last part of my talk, which will be much shorter, I just wanted to mention or talk about that synaptic homeostasis can also be maladaptive. So we're always thinking of synaptic homeostasis being as a protective mechanism. So when neuronal activity is changed, synaptic homeostasis kicks in to kind of counteract those changes and protect neurons and synaptic signaling. But in some pathological conditions, it's being found that synaptic homeostasis can also be maladaptive and even contribute to the progression of the disease. So I'll just show you a little bit of work that we've done in the past using a Drosophila model for Alzheimer's disease. These Drosophila mutants are transgenics that were created by these groups here that expressed the human toxic form of beta amyloid called A-beta 42. And it's fused to a signal sequence that directs its secretion from the cells, so to mimic sort of the extracellular accumulation or buildup of this A-beta-42 toxic protein. And in this model, these flies also have many of the hallmarks of Alzheimer's disease, including extracellular A-beta-42 deposits in the brain, age-dependent learning and locomotor defects, progressive neurodegeneration in the brain, sleep abnormalities, as well as a shorter lifespan. When we looked at these A-beta brains, and we looked at miniature EPSCs in these brains, we looked at early stages and later stages. This is in control, and this is in the A-beta-42 expressing brains. We saw that the A-beta-42 expressing brains exhibited this early hyperactivity, followed by, in the later stages, it exhibits this synaptic depression or synaptic silencing. And this is a pattern that's being found in many mammalian models, right? For many decades, it's been known that Alzheimer's leads in aged patients and models to synaptic depression or silencing. And more recently, they found that there seems to be an early hyperactivity that occurs. So we wanted to see if we could use our model to see if maybe that early hyperactivity is triggering endogenous homeostatic mechanisms and leading or contributing to the synaptic depression that happens later. So to test that, we wanted to use a lot of pharmacological agents. So we wanted to see if we could use our culture system, so go out of the brain and back to the culture system. And at early times in culture, these primary neurons exhibit the same thing, an early hyperactivity at 5 days. And this, you can see that's exhibited by a decrease in inter-event time, so much more frequent events. But if we look in culture at later times, at 9 days in vitro, we see synaptic silencing from the A-beta expressing neurons. Okay, so to look at this pathway where A-beta is creating early hyperactivity and later synaptic depression, we wanted to see if we prevented this early hyperactivity, could we prevent this later synaptic depression? So here's the 9-day-old results that I showed you earlier. So this is the synaptic silencing in the A-beta expressing neurons with much greater inter-event times. And now we block this early hyperactivity by exposing the cells to curare just from 4 days to 6 days, and then washing out that curare and recording at 9 days, just like this. And you can see here that when we do that, we prevent that synaptic silencing. So A-beta expressing neurons are now active compared to the MOP-treated A-beta neurons, suggesting that blocking early A-beta induced hyperactivity prevents downstream synaptic silencing. And the last thing that we did that I'll show you is that we wanted to see if we got rid of A-beta, could we induce early hyperactivity and see if it could lead to later synaptic depression. And we did that using wild-type flies, and then we exposed them to this drug called abinonium, which is an inhibitor of acetylcholine esterase, which is an enzyme that breaks down acetylcholine. So in the presence of abinonium, there's more acetylcholine and increased activity, and we did that just at these early stages and then recorded at 9 days in vitro. And you can see here that we do induce synaptic silencing. So we create this early hyperactivity, record here, and we mimic and get what we see with the A-beta flies and get synaptic silencing. And interestingly, when we do the same experiment in a D-alpha-7 null mutant, we don't get any synaptic silencing, suggesting, and the reason we did, of course, D-alpha-7 null mutants is because of the work I showed you earlier, that D-alpha-7 is involved in synaptic homeostatic mechanisms. So increasing early activity is sufficient to induce later synaptic inhibition, and then it's dependent on alpha-7. So in this last summary, so I show you that at synapses, when exposed to expression of A-beta-42, it leads to early hyperactivity that then triggers mechanisms that we think are homeostatic, endogenous synaptic homeostatic mechanisms that then lead to synaptic depression. And so this is an example where synaptic homeostasis can be actually maladaptive in pathological conditions. And then I'll just thank my lab. This is my current lab. I have 3 graduate students and a couple of senior staff members. These are the folks that did most of the work that I showed you today. This is Colin Hom, Yongping, and Nasser Eyadiam. And thank you for your attention, and I'll take any questions.
Thank you, Susan. Thanks. Do you have questions?
Hi, it was such a great talk. And I want to ask you if all these homeostatic changes can be then evaluated in a behavioral way, or how is this translated into functional activity of the model?
So that's a great question. And so the reason we want to move in vivo more and more is so that we can eventually relate the homeostatic mechanisms that occur at the molecular level and what happens at the behavioral level. So we haven't gotten there yet. Now we have a way to non-invasively manipulate activity, and we know what receptors and channels are involved. We know of a circuit from the giant fiber to the flight muscles that lead to initiation of flight and flight of the Drosophila, and it's completely dependent on the alpha-7 receptor. So our plans are to use that circuit and that behavior to start to look and see if we can manipulate activity, if it would change levels of alpha-7, and would it change the behavior of the flies.
Thanks.
Thanks.
Oh, thank you. Very interesting. Because you put the flies in 37 degrees, something like that.
30 degrees.
30, that is stressful for the flies.
Yeah, I think so. 37 degrees is when heat shock proteins would start to be expressed, and it would be more stressful. But 30 degrees, they can still live. They can even reproduce at 30 degrees. So, yeah, we were grateful that the mutation works at 30 degrees and not 37.
Yeah, you just mentioned something because this is you mentioned the heat shock proteins, and I just was wondering if there is a triggering of stress responses, you know, at the level of endoplasmic reticulum, and that could be reprogramming the transcriptional responses, you know, at cellular level.
That could definitely be a concern. We think we're not going to 37 degrees where we think that really induces stress, but also we are trying to think of other ways besides heat treatment that we could optogenetically reduce activity also and see if we see the same results. So it is we'd like to try to address that concern directly too. And
I don't know if there are different or several kinds of these paradigms. For example, you put the flies in 37 to analyze this plasticity. But do you know another protocol different different of the heat, or or to analyze in time course just to see, you know, if there is a lot of time, you know, in 37, maybe that the plasticity will be decreased, or maybe you need just to shot of stress, or to have an optimal plasticity response, because this is a natural response, you know, or a different protocol, you know, to see exactly how the animal is responding to environment conditions in terms of this plasticity.
Yeah, so we use 30 degrees, not 37, so we don't think it's inducing stress. And also, we are trying to develop a model where we can induce or block activity using light. So there's some channels that, like channel rhodopsin, but they're chloride channels and they're light activated. So hopefully we can light activate those channels to inhibit activity and see if we see the same things.
Yeah. I just because I'm interested also in circadian rhythms and sleep. Maybe you can avoid sleep to inhibit sleep in the flies, and this could be another protocol, you know, to analyze this plasticity
at
stuff.
The behavioral level. Yes, so actually that's the other behavior that we are also interested in sleep. So we've recently brought sleep behavior into our lab, and the goal is to look at sleep homeostasis. So inhibit sleep in the nighttime, produces an increased sleep during the daytime, and we're wondering if that homeostatic response is driven by the same pathway here. We probably won't start with that behavior because we know it's not exclusively dependent on the alpha-7 receptor, but that is the second type of behavior that we're interested in looking at.
Okay, thank you very much.
Thank you.
Beautiful talk, thank you. So my question was, so as you showed this work on the amyloid beta, and as you know, one of the few drugs available for Alzheimer's disease is this cholinesterase inhibitor. So what will be your outlook or the perspectives of the therapeutic potential of this, using this D-alpha-7 receptor as a therapeutical target, especially given the last data you showed at the end?
Yeah, I'm not sure. I think, so we're hoping that the players translate to mammalian models, but that hasn't been looked at. We do have a colleague who's also looking in mouse Alzheimer's models and trying to look at effects of the A-beta on the alpha-7 receptors and different subtypes. But yes, we would have the same interest to see if this pathway is involved there, maybe it could become targets for treatment different than affecting acetylcholine esterase. But yeah.
Thank you.
Hi. Thanks for the talk. So, first on all, the recordings are patch clamp, right? Wholesale recordings?
Yes, that's right. Perforated patch recordings.
And I was wondering that if you have patch clamp recordings, you can record the somata, right, from the cells.
Yes.
Did you check the firing discharge after all these manipulations? Or it's not important?
No, that would be a good way to look at another type of output is the firing rate, because some people look at firing rate as is under homeostatic control. One of the problems is that we do look at ax potential firing, and we've done it in the culture. But in the intact brain, what we found is that when we record from the Kenyan cells, that we saw no firing. And I think the reason is because the voltage-gated sodium channel is only found in the initial segment and not found in the cell body at all. And so insect neurons are kind of different in that they have a very round cell body, and then they have usually one or two, but often a single process that then divides into an axon and a dendrite. So it can be, we're not so sure that firing actually passes through the soma, and the firing might happen just in the axons, and which we're unable to. We've been able to patch clamp in culture from dendritic branches, but recording from the axon in the intact brain is just too difficult for us.
So you need cultures to perform firing rate analysis.
Yes, that's right. And I think because when we dissociate the neurons and put them in culture, probably some of the sodium channels are now, the membrane has shifted and it's in the.
And what about synaptic stimulation? It's not possible in the.
So we also would like, no, I think that would be great to look at evoked, right, synaptic activity.
To do that.
So I think that's our plans. So we hope to, so we can record from the Kenyan cells, and the presynaptic cells are in the antennal lobe, and they convey information from about largely olfaction, and they project to the Kenyan cells. So we can definitely have ways to selectively express genes in the presynaptic cell. So we'd like to express, for example, channel rhodopsin in them, use light to activate the presynaptic cell, and record from the postsynaptic cell. But we're not there yet. So we're just sort of moving in vivo and seeing if that phenomena is there yet. But we would love to go there.
And the last question, for this brain, you make like brain slices, in vivo brain slices, or is the whole brain, the whole?
It's the whole brain. It's the whole intact brain, just because it's so small, so we can record from it like a slice recording.
Oh, okay.
Yeah.
Beautiful. Thank you very much.
Thank you very much.
So, well, I will thank you again, and we will meet again in the first floor for the lunch. Thank you, Susan.
You.